Clear? Okay. Is the audio fine? Can someone's message?
Yes, sure, yeah, it's fine.
Okay, thanks. In the last class, we were discussing about the scientific method. That's where we were. And we discussed quickly, we'll go through it. To observe things, we should have keep our eyes open and in search for knowledge and something like that. Then we are going to see many things. We observe them closely. Then we form a hypothesis about what is its function and so on. Then we perform an experiment, and then based on the experiment, we get some results. We analyze the data and then report our findings. And as a scientist, we should also be open to um, peer review. That is about let others see what they would find it, right? What would they find if they perform the same experiment? So as I said, science is a celebration of doubt, right? It's like you are each are not just are allowed to doubt, you are actually supposed to doubt, but then we shouldn't stop there. That is the difference between a scientist and everybody else. We may have doubts, we will do the research for to clarify the doubt, proper research, and then we will see whether the information that we got is true or false. That is how scientists work. Many people say, for example, they will just have a doubt and do nothing about it. They will form an opinion already, just without doing any research on it. And that's not science. Okay. In many other things, you are not even allowed to doubt. But in science, you are supposed to doubt, but you are also work towards clarifying the doubt for yourselves and others too. So in the last, we also had this discussion about a torchlight fault, whether it is dead batteries or is it a bulb. And then based on the experiment that you replace the batteries and it may not work, then it means the batteries were not dead, probably. Or you replace the bulb and you see, and then it turns on, that means the bulb was the one that is actually burnt. You may also sometimes have, even if you change the bulb, you did not have any light. What does that mean? It is there are several possibilities where both batteries as well as bulbs are dead. Or it could be that the connection between the batteries and the light, the switch is there, maybe something faulty wiring is also possible. So we need to find multiple options. Say if this didn't work, then we should have thought about other factors that are involved in it. But as a scientist on a daily basis, we know that the usual the problems are about batteries and the bulb. Okay. So I want to talk about a small quote about from Rosalind Franklin. I hope you know who she is. If not, we will learn in the next classes. My method of thought and reasoning is influenced by scientific training. If that were not so, my scientific training will have been a waste and a failure. So that is something that you need to train yourselves as a youngster so that you will be able to do better science in the future and also better lives. So here is an assignment that I'm going to give you. Please be careful, note down whatever things are there. The answers of it is not what I'm looking for. Okay. Some years ago, I received a question paper from some African country, high school student. They have two questions here, and they both are based on how you would based on scientific thinking. Actually, how would you make a hypothesis? What kind of experiment would you do, and so on. So I'm giving you the same question paper. I will email it or post it on the Google Classroom. I want you to look into it and then make a video of you presenting. So the video should have, I will separately send you the instructions, but otherwise you can use Google Meet to record your video, and your face should be visible, and it should be based on a PPT. Okay. This is to ensure that everybody has got an opportunity to make a video and then present it. Because in the internal assessments, we are going to have 8 assessments. So out of which 5 out of 8 is what I'm going to take. All the 5 will be video-based assessments. It's not like you will be given some 7 days or so to post the video. Okay, the instructions will be given to you at that time. But otherwise, this is a trial, and I will give you a deadline to upload this, and then everybody has to do it. Okay. One thing about scientific is scientific thinking, or in general, how you identify, is some of the things you should know about psychology. That is about cognitive fallacies. We make several things. Our mind plays games on us in a way that we become biased, and then we will see what we want to see, not what is actually the truth. I'm not going to discuss about it. You can go to the link, and even on YouTube, you can try about cognitive biases, and they will give you so many ways of how we acquire knowledge and what kind of knowledge we acquire. Have you ever noticed we like the people who like the same thing as we do, right? We don't see if there is good person or a bad person. We will see if we have shared identities, then we like them more. That's kind of cognitive bias too. But those are all things that you psychological ones. You might want to go to YouTube and understand yourselves better. Okay, so this is the lesson. Nucleic acids convey genetic information, or the lecture is about it. And what we are going to focus is on some of the experiments, like Hammerling's experiment, uh, animal cloning experiments, Griffith's experiment, and every McCarthy experiment, and Hershey and Chase. There are many other experiments that might come during the course, but these experiments are about trying to understand how people have discovered that nucleic acids convey the genetic information. We will start with a brief introduction about the history, about what people thought in different times. The first one, of course, there are several theories to start with how life has started on Earth and how humans are born. The first one would be that God has created us all. That is the creationist theory. And then you have many other ways. So here is something what people understood at that time. I don't want to go into the details. So from father into the gametes, the characteristics are flowing, and it comes as heat, cold, and so on. There are a lot of things that we don't really understand. Then we will have there is another one which is called as in which people thought that this is the structure of a sperm. Supposedly there is a small minuscule human in this, and when it is impregnated into a woman, it gave rise to a new organism. That is what other people thought. And we also know about Darwin. Darwin has had his own ideas. Say, if there is an individual, and these are the gonads, say, that is where the gametes are being made, how come characteristics of skin, head, eyes, or hands, how do they come from this to the new offspring, right? So he thought something like a body, different small particles from different parts of the body, which he termed as gemmules, they reach to form the egg. And this egg, when it develops, upon fertilization, and so the same is the case with this sperm, and upon development, it forms an embryo and then an organism, right? That is his theory, because at that time, um, sorry, he wasn't aware of Mendel's work. Mendel was also kind of contemporary to Darwin, and even Darwin didn't know exact molecular ways of how things happen. So there are many other experiments in the interim that I'm not going to discuss, but we know some of the experiments had a key role in our understanding of it. In all of these experiments, I want you to remember what you have to understand: what is the model organism? Why is that the choice? What is the method used or technique, the principle of the technique? And then how do we deduce the results? That is how you have to think. In the future, if you are going to be in research, it doesn't matter if it is biology or not. It's still, there is something called as research aptitude, right? That is what you should practice. It usually comes with experience, but it doesn't mean it shouldn't come with theory. It's all about the knowledge, however you gain it. So the first question that people wanted to answer was, where do cells store hereditary information? We'll try to see a few examples, a few experiments on that. So here is uh, a moss-like thing. It's called as a stabularium. You can see that it is growing to somewhere near 1 centimeter or so, and it is single-celled, right? And although it seems to have something like roots here, there is a stalk and there is a crown here. There are three different parts and so on, but it is made up of one single cell. So that's a good model organism for this purpose. So it depends on the model organism that you choose will depend on possibilities and the question or objective that one is trying to answer. So Hammerling is a scientist who worked on this. He took two species of Stabularia. One is called as Stabularia crenulata, and the other one is called as Stabularia mediterranea. So these two, they belong to the same genus but different species. They also have different characteristics. One of the characteristics is the color. Crenulata is green in color, whereas mediterranea is brown in color. The second one is the crown, this part of it. One of it is like a bud. Crenulata is like a bud, whereas mediterranea is like a flower. So what he did was he cut it into several parts like this, and he moved this stalk region of crenulata on top of the base um, of Mediterranean. And he did the same reciprocal way. He took the stalk from Mediterranean and put on top of um, crenulata base. So what will grow on top of it is what he was looking at. Although the stalk here is brown, it has given rise to a green color that is of crenulata crown. And here, in the other case, it has given rise to a Mediterranean type, that is the flower-like culture. So one thing I forgot to mention in the previous slide is, in the base of this, there is a structure which is the nucleus. So the nucleus is not present, is present only in the base here. Okay. So what the conclusion from this is something like this: whatever color, I mean, the nucleus belongs to, the crown is of the same thing. If it is if the nucleus comes from crenulata, you will get characteristics crown characteristic of granulata. If you have a nucleus of Mediterranean, it will develop in a crown that is characteristic of Mediterranean. So what they thought is, I mean, they could also do something like this. They just they I mean, there were many other experiments done in the previous. What you should remember is, say for example, I take a, a more stable area like this, and it is the stalk. I cut it into 3 pieces, and I will see what happens. This is one part, and you have a stalk, and you have a base. This one dies away. If I just left it and see what will happen, this one died, the stalk region, sorry, the crown region died, the stalk region died. This, the base was able to regenerate into a crown. That means something hereditary is present in this, in the base, right? And second, you can also do, I mean, if you somehow cut off this part alone, nucleus alone, and the rest of the organism dies. So many times through multiple experiments and something like this, where they have done reciprocal exchange of the stalk regions, restores the characteristics that is based on the nucleus. Okay, so people have decided or concluded that the genetic information is present in the nucleus. To support something like this, if you have heard about animal cloning, um, that is popular among many people, many research areas, especially in stem cell biology. Some experiments were done a long time ago where they took an egg from a frog, and then using UV radiation, they destroyed the nucleus of it and see what happens. And then those, something like this, they died. Okay. Then they did something else. They destroyed the existing nucleus with UV light, and then took a nucleus from a different cell, and then put it into it and see what is happening. That is nothing but animal cloning, or you can call it as somatic cell nuclear transfer. We will get to those technical terms later. So when this kind of nucleus transplantation happened, right? You took a nucleus from a different cell and put it into an egg, a nucleated egg. That is, you destroyed or removed the egg. Majority of them would have died, mostly because we are doing something serious manipulation. So there is always a chance for DNA damage. Some of those kinds of manipulated embryos, they manipulated eggs, they grew to certain stages of embryo, and then they died. The growth was abnormal. But very rarely, what happened was the embryo developed into a tadpole and grew into a complete organism. Now, I gave you two situations. One is that when you just remove the nucleus, as I showed here, if you remove the nucleus, the egg is dying. But if you are replacing with a different nucleus, it is growing into a complete organism. Again, this is the conclusion that the genetic material is present in the nucleus. Okay. But the problem is nucleus has many things. If you learned in biochemistry, you would have learned that these are 4 biomolecules. You have proteins, nucleic acids, lipids, and carbohydrates. A nucleus has all of it. Lipids, as in the nuclear membrane, it has several proteins, say for example histones, and nucleic acids, you have DNA and RNA in it, because transcription happens in the nucleus. DNA is the genetic material, and there will always be some carbohydrates here and there attached, probably. So people had now we have to decide what is the chemical nature of the genetic material. Okay, we know four things, out of which you have proteins, nucleic acids. In nucleic acids, you have DNA and RNA. Then we have carbohydrates and lipids. So there is a big reasoning about why people thought that carbohydrates and lipids cannot function as genetic information. For a molecule to perform the function of genetic information or be as a genetic information, there should be diversity and it should be long. That is, it should be a polymer. And there should be a diversity in the molecules. If you know about carbohydrates, say glycogen or starch, they are polymers, but they do not have that much diversity. The subunits either could be glucose, all of them are glucose. If you take a little more complex, they may have at the most two types of monosaccharides. So that's not so much of a diversity. So carbohydrates cannot function. That's the logic in general you can think of as genetic information. Lipids, they do not form polymers, right? You will see in the membranes as something like this. They are arranged, but they are not polymers. So carbohydrates and lipids are excluded, that they cannot be genetic information. They cannot act as genetic information. Of course, through experiments too, people have proved that the carbohydrates and lipids cannot. Then we are left with proteins. They are polymers of amino acids, and we have 20 different types of amino acids. So there is diversity, and there are also polymers, long. Nucleic acids are, you have two different types, DNA and RNA. DNA is a polymer, but it only has 4 bases. RNA is also a polymer, but it also has only 4 bases, subunits we are talking about. So majority of the people believed at that time that proteins are likely because they have large number of amino acids. So there is a huge diversity. So proteins are likely to be the molecules of heredity. So at around 1940, 50s, there was some nice experiment done that is called as by Griffith, and we call it as transforming principle, because he has shown something which we will discuss now. He worked with 2 different strains of Streptococcus pneumonia. When I say strains, I hope you understand. Streptococcus here is genus, right? Pneumonia is species. And within them, there are further divisions, and those are referred to as strains. So here is R strain, all these small ones you see, and here you see the big ones. Both of them, these are what you're seeing here are colonies. Okay, what is a colony? One bacterium is there, and it will divide and make one. That is one colony. Okay, so one of this part represents billions of bacterium in there. So R strain, there are few differences that we need to know. R strain and S strain. R strain forms small colonies, and they form rough colonies. That's why R is for R. They call it R strain, which is called rough strain. In this one, I hope you see that the surface of the colony is kind of smooth. That's why they called it as S strain. The difference between if you look at one cell, okay, you take out one cell from here, they both are almost same size. From one cell from here, they both are same size, same everything, except there is one difference. The difference is the smooth one has a polysaccharide capsule, and it is smooth because it has a capsule on surface of it. Whereas the rough one does not have this, it does not have the capsule, so it forms rough colonies. Okay, so that's the difference. And one more thing you need to remember is, I have a cell bacterium like this and a bacterium with a capsule. Which one do you think will have more protection? Is it the rough one or the smooth one? Say if R strain enters into a body, what will happen? The antibodies are produced and the antibodies will kill it very easily. Whereas the smooth strain will not die that easily because the capsule that is there, the capsule protects it from being attacked by the host immune system. So the S strain can cause a disease, that is pneumonia. Whereas R strain does not cause a disease because it is eliminated by the immune system. So now we are going into, I have described the model organism, and there is little bit of genetics to some aspects of microbiology. That's why I explained those. Now let's see the experiment itself. So again, we should recollect what I have told you. R is non-virulent. Why is it non-virulent? Because it does not have the capsule. Therefore, it is attacked by the immune system and then will be eliminated. Whereas the smooth one is virulent. Virulent, when you say, it means that the ability to cause a disease. The smooth strain, S strain, can cause disease because it has a capsule and is protected from being eliminated by the host immune system. So here we are going. Here we are taking the S strain. This is the capsule in blue color. This is S strain. He injected into mice, and the mice caught pneumonia, and the mice died. Okay, what are we showing here? We are showing that S strain is virulent. It can cause disease. Now we are taking, this is the R strain. Okay, it does not have the capsule like in the S strain here. You inject R strain into mice, mice do not die, they are alive. So R strain is non-virulent. That means it cannot cause disease. Why doesn't it cause disease? The antibodies of the mouse will eliminate this R strain. So mouse is safe. Now we are doing something different. You take S strain and then you heat kill it. That means you boil the culture of bacterium with S strain, and now you take those killed ones and inject into mice. The mice is alive. What does it say? Normally, S is virulent, right? But heat killed S is not virulent. Okay. And you also, we will write this. R strain is non-virulent, right? The next one is what, if you understood correctly. We will review this experiment again. Don't worry if you're lost somewhere. Now, what he did is, okay, if you give R strain by itself, it is non-virulent, right? If you give heat killed, this is actually killed one, okay? Like in this case. S, if you give, this should be non-virulent. This R is non-virulent, and S is non-virulent, R is non-virulent. You mix these two cultures and give it to mice, and the mice dies. That's something of a surprise, isn't it? So why would they would die? They would have died because something caused the disease. Which of them independently would have caused disease? R strain does not. Okay, you should also remember that R is the only live one. The S is heat killed, right? live R strain is non-virulent. Heat killed S strain is non-virulent. You mix these two and give, and the mice is dying. And they have tried to isolate what bacterium it is, and they have isolated only S strain. But S is dead. The one that we have here is S is killed. Only the live ones that we have given is R. So there is only one possibility. R must have transformed into S, and that S would have become virulent. Okay, I'll summarize it in this slide all together. You can use kind of deduction here. So S strain, death. S will give death. R strain, alive. That is, R is non-virulent. You heat kill S strain, then it is alive. Mice is alive. That means it is non-virulent. But when you mix heat killed one and live R, then the mouse is dying, and all of them are S strain. So the conclusion is something like this. From what Griffiths understood at the time, something has transformed R into S. That he called it as, he doesn't know what is it. I mean, the chemical nature of it. So he called it as transforming principle. Okay, Griffith, this is what the experiments performed by Griffith. Now, because we know a lot of things, that then we will say what is happening. So S strain has DNA. The difference between R strain and S strain is only one gene, at least one important gene, that is called as CAP-S. That gene produces an enzyme that will synthesize the capsule. In the R strain, this is the R strain. Okay, this is the S strain. You see the capsule here, that one. R strain doesn't have a capsule because the gene here, it got mutated, and it doesn't produce the enzyme which will synthesize the capsule, or which is required for the capsule formation. So when we have this heat killed, the cells are broken down, the DNA is broken down into several pieces, in one of which the CAP-S gene, this is the functional gene, okay? The R strain has a non-functional gene. So the CAP-S gene has been taken up by the rough strain, R strain, and recombination occurs. So the CAP-R is replaced with CAP-S. That is homologous recombination. We will read those details later. So there is homologous recombination. The CAP-R is now replaced with CAP-S. So because this R strain now has a functional enzyme to produce the capsule, it has become S strain. This was actually R strain, but now it has become S strain. Okay. So that is what our modern interpretation. At that time, nobody knew about the genes or how these mechanisms happened. So the conclusion of Griffith's experiment is something is called as the transforming principle. There is some part within the bacterial cell that can transform a trait from one cell to the another cell. Okay, and that is because so R is transforming into S, so that is called as transformation. Okay, so we still don't know the chemical nature of it. Therefore, there were next set of experiments were carried out by Avery, McLeod, and McCarthy, three important scientists. They are kind of the first people to show that the the chemical nature of genes or genetic material is DNA. They performed a nice set of experiments. They have taken the same model organism and kind of material also. I mean, the procedure also is almost the same, except some difference. They took heat killed cells, and you have the whole mixture. And now they have added proteinases. What will proteinase do? It will degrade all the proteins. And in one of the other tubes, they added ribonuclease, or you can say RNase, that will degrade all the RNA. And in a third tube, they have added deoxyribonucleases or DNases that will degrade DNA. And now, after treatment with these enzymes, so normally, if you take in this tube, it had carbohydrates, lipids also. But as we discussed in the previous slide, they are not suitable molecules, and so they were eliminated. What we now have is proteins, DNA, and RNA. One of it is the genetic material, or is the transforming principle, and that is what these scientists wanted to find out. So I will just put it as PDR, protein, DNA, and RNA. I will write for all the three. P, D, sorry, D, R. So because we added proteinases, we deleted the proteins. And upon adding to R culture, what has happened? There were S cells in the culture after growth. So I hope you understand this. They take a sample of this, and then they will add it to live R cells and see if they could find S cells or not. And they have found S cells. So protein protein is, or pre is not the protein is not the limiting factor for the transformation, right? In RNases, in the second tube, they have added RNase. So that means I will remove this. Still, they observed that R has changed to S strain. In the third one, they have removed DNA, and then there is no S strain. They would only found R strain. What does that mean? Um, how you interpret it is important. You can say in this one, D and R, okay, DNA plus RNA are required for. I'm just making it as a complex one. I hope you will see it as we will get to it. Transformation. What else is found? Protein is not required. That is what the information you get from tube 1. Right? DNA and RNA can, if they are present, transformation occurs. Protein not present, transformation still occurs. In the second tube, you have eliminated RNA. So protein is present. You can still see transformation. RNA is not required for transformation. That is what you're saying. Here you're saying protein is not required for transformation. In second tube, RNA is not required for transformation. DNA is removed. RNA plus protein, and DNA is removed, and no transformation. What this one says a lot. RNA and protein cannot by themselves mediate transformation. If DNA is not there, transformation does not occur. So by deduction, you can say that DNA is required for transformation to occur. Okay, so DNA must be the genetic material. That is how people do the experiment. If anybody had a question on this, you are welcome to ask. Don't feel shy about it. if you have any questions, please interrupt me. Any questions here?
Sir, I have
Yes.
one doubt, sir.
Yes.
I think we
Yeah,
have the same term.
Aditya,
yeah,
who
I'm
is,
pretty sure you have the same question.
which one is it?
Uh, why were carbohydrates and lipids initially eliminated? What was the
Uh,
primary reason for it?
okay, I'll try to go back. I'm unable to do that, I think. Okay, I will. mm, So carbohydrates, if you know, for any information to be there, it should be long. That means it should be a, a polymer, right? And
How did they determine that, sir?
say it again.
How did they determine that if there's any information, it should be a polymer?
Okay. What it means is it should be one molecule, one long molecule. Why is that? It is because based on the number of characteristics. You have so many characteristics and there are so many molecules, proteins, and enzymes. People knew about enzymes and the composition in a broader view. If something that is little bit philosophical, it goes into information. If you want to see any kind of information, and if something like a complex organism, complex something like life, a cell, it has many molecules, say 10,000 molecules. If a genetic material is very small, it is unlikely that it is going to code for 10,000 molecules. Okay, so the information of these 10,000 molecules should be within the genetic information. If that is the case, then the molecule should be long to hopefully have. Now, when in my answer, I'm trying to put it as because we know it now, that DNA and so on. Otherwise, if carbohydrates are there, for example, in no carbohydrate, you would see a variety in the sequence. You may see a starch or something like that. It is a unit of glucose, polymer of glucose. If from the start till the end of the molecule, if it is the same subunits, okay, how can it code for diverse molecules?
Okay, there's no room for diversity.
Yeah, only if there was some difference in the sequence, then you would have diverse molecules. Otherwise, you will just have 10,000 molecules of one particular protein, say, for example.
Yes, so that makes sense.
You need at least 10,000 different types of proteins. Okay. So number of characteristics and the diversity of characteristics will determine, or if you base on those, then you can logically come to this, that it should be long and it should have diverse subcomponents. Did I explain?
Yes, sir.
Yes, sir. Yes.
Okay. Any questions? Any others? Okay. If you don't have questions, I'll move forward. And I have one more experiment to describe, and then I'll stop. So the other experiment is by Hershey and Chase. They worked on viruses, or you call them as bacteriophage. Maybe I will just introduce um, the experiment, and in the next class, I'll repeat this experiment and we'll complete it. So bacteriophage, phage, I hope you know that's eating, right? Those that eat bacteria were called as bacteriophages. They are viruses. I hope you know that. Anybody can say what are um, mycophages? What do you mean by mycophages? Anybody? Myco is for? What is mycology? Can you recollect?
So fungi.
That's right. These are viruses of fungi. They are referred to as mycophages. I think there are also phycophages. Okay? Anybody?
Phycos for plants.
No, FICO, PHY.
But phyc,
This
uh,
is for, yeah, you want to try? Algae. Okay, algae, viruses of algae are called as phycophages. You have plant viruses, animal viruses. You can take any kingdom in living things. You have those respective viruses that will feed on them. Okay, so by this time, this is much more simple to work with, and that's why they used it as model organisms. Viruses have a protein coat, and they have either DNA or RNA, either of them typically. But if you look at all the bacteriophages, they only have proteins and DNA. Uh, Animal viruses and some others have RNA as genome. But these bacteriophages, they have only protein or DNA. So one phage, one bacteriophage can infect an E. coli or bacterial cell, and they form thousands of phages, and they all look the same, right? The identity of the bacteriophage, I mean the characteristics and so on, they remain the same. How is it possible? That means something, there is some genetic information. And what is the chemical nature of the genetic information in these bacteriophages was the question. And it's easy and good to work with it because it has narrowed down to it. Only protein and DNA are present. So they have done an experiment called as blender experiment, because they used something like a mixie at our homes, and they did. I will describe this in the next class because don't think I have sufficient time for it. But otherwise, you would also know the chemical nature. I'll give you some introduction to the experiment so that you will understand for a better thing. What are the atoms that you will see in proteins? What are the atoms that you'll see in nucleic acids? I want some answers. Anybody? C, carbon, is it there in proteins?
Nitrogen,
Yes, nitrogen,
hydrogen,
hydrogen.
um, oxygen,
That's right.
oxygen, a sulfide, for sulfide bonds.
Yes, some of them sulfur-containing amino acids are present, right? Some of the amino acids have sulfur. Can anybody name those?
Methionine.
That's right.
methionine.
And
Cysteine, sir.
that's right, cysteine. So disulfide bonds form because of cysteine amino acids. Okay, in nucleic acids, what do we have?
Again, carbon.
C, H, N,
Phosphate, sir.
O, and P,
Oxygen.
right? If I want to particularly label proteins, and which atom should I choose? I cannot choose carbon atom because it is also present in nucleic acids, right? So which atom should I choose to label? Label means I want to put some radio labeled atom.
Sulfur.
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 That will be sulfur. If I want to specifically label nucleic acid, then what would be the atom? That is phosphorus. You do not find phosphorus in proteins. You do not find sulfur in nucleic acids. So that is some of the experimental setup, which we will describe in the next class. So as we said, proteins have sulfur. The bacteriophage has something like this structure. The outside one, you see the hexagonal, or that is made up of proteins, and the inside one is made up of the DNA is inside, like is shown in this figure here. So if you have, they have taken two different sets of phages. In one set, they have labeled it, labeled only the proteins. So they would have used S35 radioactive radio label. And they have used, for the second set, they used P32 radioactive atom. How would they label them is again a huge experiment by itself. What they would, I would only take it there and I will stop, so that the experiment itself we'll learn later on. So what they would do is, they will take a bacterial culture and they would add bacteria to grow. It needs a lot of nutrition, right? And you will add glucose, you will add a nitrogen source, you will have a carbon source, you will also add some phosphates and so on. And you will also add some sulfates. So these three will be same. Now, if I want to label the phages, the bacteriophage, I want to label only the protein. So I would take a component, some sulfate, say MgSO4, which has a radio label there. Okay? And the bacterium is made to grow on this nutrient mixture, which has a radioactive sulfur atom in it. And then you would also, at the same time, you will also allow the phage to grow on the bacterium. So all the DNA that is present, all the sulfur containing atoms in the bacterium would have been radio labeled. And the proteins of the phage that it has to make, like I'll go here. Okay, sorry. So if the cell has, now, as I said, this is growing on MgSO4 with the radioactive one. Okay. Say if it were growing, the protein should be made from this DNA, from the phage DNA. And if it has to be done, they have to use only the sulfur sulfate that is with the sulfur radio label. So all the proteins that are being made would have been radio labeled. Okay, I hope you understood how labeling is typically done. If we want to do it for DNA labeling, then we would have taken a culture and we would have added everything, glucose, nitrogen source, and MgSO4 normal. We will add phosphate, some phosphate which is having a radio labeled phosphorus. So any nucleic acid that is being made in this culture would have had the radio labeled phosphate. So you have the culture of bacteria which is having radio labeled phosphates, and if the virus infects and makes more copies of its genome, then all of it will be labeled with radio labeled phosphate P. Okay. So I'll stop here. In the next class, we will discuss about, we'll continue from here. We'll have a quick review. I want you to read this one thing. Okay. Again, from Rosalind Franklin, science and everyday life cannot and should not be separated. The way we think, the way we acquire knowledge, the way we know what to believe or not, is not different from how and what we are learning every day in class. Okay, science and everyday life cannot and should not be separated. You have to follow scientific method in many, many things. Okay. Any questions? If not, we'll meet tomorrow morning. I hope the computer will hold on like it did today. But otherwise, I think things will go smooth from Saturday onwards. Okay. Thank you very much.
Thank you, sir.
Thank you, sir.
Bye-bye.
Thank you, sir.
Thank you, sir.
Yes, have fun.
Thank you, sir.
Thank you, sir.